Let's discuss the first MCQ. What is the order of size of the cell components? So if you recall from the sizes, so which of the following is the largest and which of the following is the smallest? If we compare mitochondria, nuclei, ribosomes, and starch grains, so nuclei are the largest, right? Then comes the chloroplasts, then mitochondria, and then ribosomes. We discussed the mnemonic NC, L, NC, MLR, right? So from largest to smallest, nucleus, chloroplast, mitochondria, lysosomes, and ribosomes, right? So the answer is B. If we talk about MCQ number 2, the diagram shows the structure of a typical plant cell. Which cell component is also present in prokaryotes? So this is a typical plant cell, which is actually a eukaryote. And we have to state that we have to choose the option which shows the structure that is also found in the prokaryotes. So D is the nucleus, and nucleus is not found in the prokaryotes. It's only found in the eukaryotes. C is the sap vacuole. It's found in the eukaryotes only. What about B? B is the chloroplast, found in eukaryotes only, the plant cells, whereas A is the cytoplasm, which is found in the eukaryotes and prokaryotes. Okay, MCQ number 3. The diagram shows this ultrastructure of eukaryotic cell. What is meant by ultrastructure? Ultrastructure is the structure of a cell as observed under the electron microscope. So this is the ultrastructure of the eukaryotic cell, and the cell shown is the animal cell. So which organelle does not contain nucleic acid? So what do you think? A is the nucleolus, and nucleolus is the site of
A.
Right. A is the answer. Exactly. Because A is a Golgi apparatus, and we discussed that Golgi apparatus does not contain nucleic acids like DNA or RNA. But if we talk about D, D is the nucleolus, and in the nucleolus there is DNA and RNA. If we talk about mitochondria, that is C. So mitochondria also contains DNA and RNA. And what is B? B is RER. So RER contains ribosomes, and ribosomes contain RNA. So A is the right answer. Okay, let's move on to MCQ number 4. Which components are present in prokaryotic cells? So we just need to state the answer that contains the components found in the prokaryotic cells. So what do you think? The answer is D?
D.
Okay.
D for dog.
Right. Exactly. Cytoplasm is present in the prokaryotic cell. Also the DNA and ribosomes. Mitochondrion is not found in the prokaryotes. Nuclear envelope and mitochondria are not found in the prokaryotes. Even chromosomes are not found in prokaryotes. Nuclear envelope is not found. Chloroplasts are not found in the prokaryotes. Okay, MCQ number 5. What identifies a cell as a prokaryote? The DNA is associated with protein? No. Prokaryotes have an... Okay, let's see. Exactly, the DNA is in a circular form. Right, this is the answer. In the prokaryotes, DNA is circular, and DNA is not associated with histones or proteins. DNA is simply naked, not associated with any protein. The DNA is in the form of a double spiral. So the DNA is in the form of double spiral in both prokaryotes and eukaryotes. This is right for the DNA, but by looking at the double spiral DNA, we cannot say that it's a prokaryote, because DNA is always double spiral or two-stranded or double-stranded, right? The DNA is surrounded by a membrane system. No. DNA is surrounded by a membrane system in eukaryotes. So MCQ number 6, a lysosome measures 0.4 micrometer in diameter. What is the diameter in nanometer? So how do we convert it into nanometers? By multiplying by
1000.
1000. So what will be the answer? 400 nanometers. That is C. Let's move on to MCQ number 7. What is the function of the smooth endoplasmic reticulum? We discussed that the function of the smooth endoplasmic reticulum is the synthesis of steroids and the transport of steroids. So what do you think is the answer? C. Synthesis of steroids and transport of steroids. Right? Steroids are the lipids which are derived from cholesterol. And steroids are actually lipid hormones like the testosterone, estrogen, and progesterone. Okay. MCQ number 8. So this is a very good MCQ. It says when mitochondria are extracted from the cells for biochemical study, they are usually kept in a 0.25 mole per dm cube sucrose solution. Why is the sucrose solution used? Okay. So what they are saying, that when mitochondria are extracted from the cells for biochemical study, they are kept in the 0.25 mole per dm cube sucrose solution. Right? So what could be the answer? To act as a solvent? No. If we are placing mitochondria in a solution, so mitochondria are the whole organelles, right? They're not a molecule that will dissolve. Okay, let's see. A is not the correct answer because mitochondria do not dissolve. They are the whole organelles and not the molecules. To enable the rate of respiration of the mitochondria to be determined. No. To find the rate of respiration in the mitochondria, what do we need? We need to measure the uptake of oxygen per unit time, and we need to measure the volume of carbon dioxide produced per unit time to find the rate. This is not the correct answer. To prevent the mitochondria from changing its structure, this is the right answer. Why? Because why not D? Can you tell me why not D? To provide a source of energy. It's a sucrose solution, right? So sucrose is a carbohydrate. Why not D?
Yes, sir.
Why not D? Why only C?
Because I guess we're not providing it with oxygen.
Over here, they have mentioned that they're using a solution of 0.25 mole per dm cube sucrose. What does that mean? They're using a specific concentration. Because 0.25 mole per dm cube sucrose solution has a specific concentration, so it has a specific water potential. And maybe this water potential is equal to the water potential of the mitochondria. So there are isotonic conditions. And what will happen? There will be no net movement of water. So mitochondria will not change in structure. Are you getting my point? If they had to provide a source of energy, they could use any solution of sucrose. They could use any concentration of sucrose, right? If they need to provide energy, they can use other carbohydrate as well. They can use glucose. They can use any concentration of the solutions, but they're just using 0.25 mole per dm cube sucrose solution. Why? To prevent the mitochondria from changing in structure, because this solution is isotonic to the mitochondria. Mitochondria and this solution have the same water potentials. So that's why C. Did you get it?
Okay, sir.
MCQ number 9. The diagram shows the ultrastructure of a typical animal cell. Which structure synthesizes and transports lipids and steroids? So lipid synthesis and transport is the function of the smooth endoplasmic reticulum, which is C. SER. What is A? It's Golgi apparatus. What about B? It's rough endoplasmic reticulum or RER. What about D? It's mitochondrion. Okay. Let's move on to MCQ number 10. Which structures are found in plant cells but not in animal cells?
D.
Okay. So centrioles are found only in animal cells. They're not found in plant cells. We have to choose a structure which is found in plant cells, but not in animal cells. Mitochondria are present in both. Nucleoli are present in both, whereas plasmodesmata are only found in plants. So I told you P for plasmodesmata, P for plants only, right? MCQ number 11.
Yes, sir.
What is the correct order of the size of the organelles? So nucleus is the largest, and then comes the mitochondrion, then comes lysosome, and then comes ribosome. Again, that mnemonic is followed: NC, M, L, R.
C.
Nucleus, chloroplast, mitochondria, lysosomes, and ribosomes. So answer is C. Okay, MCQ number 12. A piece of mammalian tissue was homogenized and centrifuged. The biochemical activity of 4 subcellular fractions was investigated, which diagram indicates the fraction with maximum synthesis of messenger RNA. So we discussed that messenger RNA is synthesized during transcription. When the genes are transcribed, messenger RNA are formed, right? Genes are in the nucleus. DNA is in the nucleus. And what is transcription? Transcription is when the gene is copied, right? And messenger RNA is formed. This is transcription and this occurs in the nucleus. So what do you think? What should be the answer? The answer should be B.
Can I
Why?
answer?
Sorry. The answer should be B. Why? Because the nuclei will have the most activity of synthesizing the messenger RNA, because messenger RNA is synthesized in the nucleus during transcription. Transcription also occurs in the mitochondria, but more transcription occurs in the nuclei, in the nucleus, as compared to the mitochondria, right? Mitochondria synthesizes its proteins as well. Mitochondrion has a circular DNA, and that is also transcribed. It contains genes which are transcribed too, but most transcription occurs in the nuclei. So the answer should be B. Is it okay? Fine.
Yes, sir.
Okay. Let's move on to MCQ number 13. The diagram shows a drawing of an electron micrograph of a cell. So this is a drawing of a picture taken from electron microscope, and the picture is of a cell. This is an animal cell, which structures are surrounded by double membrane. So we discussed that Golgi apparatus is a single membrane bound organelle. It's not surrounded by a double membrane. Same goes for lysosome. However, mitochondrion is surrounded by a double membrane. It's a double membrane bound organelle. So is the nucleus. And cytoplasm is not surrounded by a double membrane. Cytoplasm is surrounded by a single membrane, that is cell surface membrane. So the answer will be C. Okay. Right. Let's move on to MCQ number 14. In which animal cells would Golgi apparatus be most abundant? So I told you that the cells which are synthesizing and packaging proteins, or we can say the cells which are secretory cells, which are secreting proteins or glycoproteins, they will contain abundant or a lot of Golgi apparatus. So what do you think should be the answer? The goblet cells are secreting mucus, which contains what?
Yeah, I think it's B.
You think it's D or B?
No, sir. B for ball.
Okay, good. Right. So goblet cells secrete mucus, and mucus contains glycoproteins. So for the synthesis and packaging of glycoproteins, Golgi apparatus are required, right? That's why Golgi apparatus will be most abundant in the goblet cells. Okay. MCQ number 15. Which adaptation would increase the efficiency of active transport of carbohydrates from a plant cell? Okay. So what do you think? Which adaptation would increase the efficiency of active transport of carbohydrates from a plant cell? Areas where cell wall is thin, increased permeability of the cell wall, large surface area of the cell surface membrane, selective permeability of the vacuole membrane. Okay, so you need to know, basically this MCQ is from the cell membrane chapter. It's linked kind of. It's linked with the cell structure, and basically it's the MCQ from the cell membrane chapter. So let me tell you that for active transport, what are required? Protein pumps are required for active transport. right? These are the proteins in the membrane which pump the molecules or ions from a lower concentration to higher concentration in active transport. Are you getting my point? And these protein pumps are found in the membrane, in the cell surface membrane. So what would increase the efficiency of active transport? More protein pumps will increase the efficiency. And for more protein pumps, there should be large surface area of the cell surface membrane. Are you getting my point? If cell wall is thin, it doesn't matter. Okay. If cell wall is thin, it doesn't matter because cell wall allows increased permeability of the cell wall or selective permeability of the vacuole membrane.
Sir, D.
D for dog. Okay. Why it shouldn't be D?
Yes.
Because active transport occurs across the cell surface membrane, right? It rarely across the vacuole membrane. And even if the active transport occurs through the vacuole membrane, so what has selective permeability to do with the active transport? We are right now talking about the efficiency of active transport. Always remember this point, that efficiency of active transport is increased by the large surface area of the membrane and more protein pumps. Obviously, when there will be large surface area of the cell surface membrane, there will be more membrane. Do you get this point? Whenever there is large surface area, there is longer length of the membrane, and there can be more space and more protein pumps will be accommodated. So more active transport will occur. Any other point that can increase the efficiency of active transport? More mitochondria, more ATP. Are you getting my point? So whenever they say if a cell contains a lot of mitochondria, so the cell can carry out a lot of active transport because it will be producing a lot of ATP. So what do you think the answer should be? C.
C.
Which is a feature of all prokaryotic cells? Absence of cell surface membrane? No. Cell surface membrane is present in all prokaryotic cells. Division by mitosis? No. Prokaryotes divide by binary fission. They don't divide by mitosis because they don't have a nucleus. Presence of mitochondria? They do not have mitochondria. Presence of ribosomes? Yes. Ribosomes are found in all the prokaryotic cells. And can you tell me what type of ribosomes? What type?
70S.
Excellent. 70S ribosomes are found in the 70S ribosomes are found in the prokaryotic cells. MCQ number 17. The diagram shows an electron micrograph of a typical animal cell. What is the function of the membrane system labeled X? So what is X? Can you tell me? It's yes.
Rough
endoplasmic reticulum, RER. Okay. So what do you think is the function of the rough endoplasmic reticulum? We discussed that the function of the rough endoplasmic reticulum is protein synthesis and transport. So what do you think is the best option?
D.
Okay, let's move on to MCQ number 18. The diagram shows an electron micrograph of a plant cell. What do the structures X, Y, and Z contain? So can you tell me what is Z? Z is the nucleo.
Sir, what letter do you want?
Sorry, Z. So Z is the nucleolus over here because this structure is the nucleus, right? This is the nucleolus. What about Y? This structure is the chloroplast. Wait, let me highlight it with a different color. This structure is the chloroplast. And what do you think?
Starch.
Very good. So Y is the starch grain. Okay. What about structure X?
According to the table, it's mineral ions.
Okay. We will move on to the table later on, but I want to know what is actually X.
Sir, I think it's vacuole.
Very good. It's actually cell sap. And exactly, cell sap is in the vacuole. Very good. So if we highlight the vacuole, this is the boundary of the vacuole or the tonoplast, we can say, right? So what do structures X, Y, and Z contain? X is the cell sap. So it contains water. It contains mineral ions as well. What does Y contain? It contains starch because Y is starch grain. And what about Z? Z is the nucleolus. And we discussed in some previous MCQs that the nucleolus contains DNA and RNA. Nucleolus is basically a loop of DNA. And in the nucleolus, ribosomal RNA is made, or ribosomes are made. So nucleolus contains RNA as well. The answer is D. Okay. Which organelles are found in cells of both eukaryotes and prokaryotes? So
D.
ribosomes are found in both eukaryotes and prokaryotes. Chloroplasts are only found in eukaryotes, and same goes for Golgi apparatus and mitochondria. The answer is D. Let's move on to MCQ number 20. The diagram is taken from an electron micrograph of a cell which secretes digestive enzymes. Where are these enzymes made? So what do you think where the digestive enzymes are made? Digestive enzymes are proteins, and proteins are made in the ribosomes, or we can say rough endoplasmic reticulum. So what do you think should be the answer? It should be C, because C is rough endoplasmic reticulum. What is D? Nucleus. What is B? Golgi apparatus. And what do you think is A? A is basically secretory vesicle, right? Which is getting secreted out of the cell.